Some cases seem unsolvable, throwing up more questions than answers, leading us down blind alleys only to reach a dead end. But all these mysteries can be unlocked, all thanks to a single wonder molecule, nature's detective, DNA. But how does this double helix contain the secret code to all these case files? That's a question of science. Detectives have always relied on biology to fight crime. In 1860, William Herschel, a British administrator in India, first used human fingerprints to stop frauds in pension funds. In keeping permanent records of pensioners, he realized that each fingerprint was unique to the individual and remained unchanged through his or her lifetime. And so fingerprints became trusted investigative tools. In the 1900s, another forensic tool was introduced when Dr. Karl Landsteiner showed that different individuals could possess different blood types. This radical discovery prompted detectives to use blood samples to identify suspects of crimes. But none of these tools were foolproof. Fingerprints could be wiped out or prevented by a simple pair of gloves, and two unrelated individuals could have the same blood type, leading to inconclusive evidence. The question then arose, was there a biological material that would leave forensics detectives with nearly zero doubt? For the answer, they had to go deep into the living body, into the nearly 100 trillion living cells that make up a human being. Inside the cell is the nucleus, containing 46 chromosomes, 23 from our fathers and 23 from our mothers. These thread-like chromosomes are carriers of DNA. And it is here that modern-day detectives can find solutions to their mysteries. Deoxyribonucleic acid, the most powerful molecule in the biological world. Found inside every living cell, it carries the blueprint for every life form. Be it a plant, an animal, or us humans, DNA is the common denominator. The complex molecule of DNA is made of just 4 building blocks or nucleotides. They're named adenine, cytosine, thymine, and guanine. Like partners in crime, these nucleotides exist in pairs. A and T are complementary partners, as are G and C. Together, they form the rungs of the ladder that make up the double helix of DNA. There are over 3 billion base pairs in the human genome itself. That's about half the human population of the entire planet. So what makes this double helix the blueprint of all life? Just like letters in the alphabet combine to form all the words of a given language, these nucleotides or base pairs arrange themselves into genes and non-gene sequences. Genes are fragments of DNA that are responsible for a particular biological function or trait. For example, they determine characteristics like how tall we are or how thick our hair will be. Us humans possess nearly 30 to 40,000 of them, but this only makes up 0.01% of our DNA. The remaining 98.8% is non-coding DNA, or sequences that do not directly correspond to any biological function or trait. For a long time, it was believed that each of us possesses the same arrangement of genes. The idea that it could be different in different people was not common knowledge.
It was believed that all of us have the same genes. There is no difference between different individuals.
But all that was set to change, and it would inadvertently revolutionize forensic science. How? The key is polymorphism. Polymorphism is a common phenomenon in the natural world. It describes biological variations that are common within the same species. For example, all humans have blood, but this blood shows polymorphism. That is, it exists in different blood types. No single type can be designated as the standard. Each one, whether A, B, or O, is an acceptable variation. It turns out that DNA also exhibits such polymorphism. And just like blood types, its variations are inherited from one generation to the next. In the early 80s, this startling discovery had geneticists asking, was there a way to observe these variations? Yes, as was discovered by Dr. Lalji Singh, an Indian scientist at the University of Edinburgh in United Kingdom. While studying the sex chromosomes of snakes, he stumbled upon an intriguing sequence of non-coding DNA, or DNA that doesn't directly result in a biological trait. This sequence of base pairs seemed to repeat itself again and again along a specified stretch of DNA. Thanks to this property, it was called simple tandem repeats, or STR. For example, this segment of non-coding DNA contains an STR made of nucleotides GC, repeated 15 times. Another STR shows the nucleotides GCT, repeated 11 times. The fascinating fact is that just like blood types differ from human to human, such STRs also show polymorphic variation. This was an exciting discovery, but its full implications would only be understood in a lab not too far from Dr. Singh's. The year was 1984. Sir Alec Jeffreys had a eureka moment at the University of Leicester in England. He had observed STR variations in DNA samples collected from different members of the same family. These STRs showed variations in different family members. They also showed some similarity. But what did all this mean? It meant that two individuals could be told apart just by studying their DNA samples. Sir Jeffreys proposed that DNA could be used like a fingerprint to conclusively identify an individual as distinct from all other individuals on the planet. Not only that, DNA polymorphism was hereditary, so one could determine parenthood by studying DNA fragments. Sir Jeffreys' discovery revolutionized the world of forensics, and it shone a light on DNA's powerful potential to be a detective. Today we know that humans only differ from each other by 0.1%, and it's this minute difference that provides the clinching evidence.
0.1% may look like a very small number, but in terms of the actual number, it is more than 10 lakh differences.
But perhaps the biggest advantage of DNA over traditional fingerprints or blood samples is its inherent resilience.
It's such a powerful tool, DNA. Is very important because unlike protein, it's very stable. You can boil it, treat with alkali, both the strand of DNA will come apart. The moment you put in neutral buffer, snaps back again, it gives you double-stranded DNA.
1987, back in India, the Center for Cellular and Molecular Biology had caught wind of Sir Jeffreys' revolutionary new findings. Founder-director Professor P.M. Bhargava saw a direct link to Dr. Lalji Singh's discoveries with the snake called banded krait.
He asked me, why don't I develop DNA fingerprinting probe? I was reluctant, saying that the discovery of DNA fingerprinting has already been done by Alec Jeffreys. What is there to develop? He said his probe is patented, is not available. It's going to have a tremendous application.
What exactly was this probe? Dr. Singh had identified STRs, or simple tandem repeats, in the DNA of the banded crate. This STR, GATA, repeated itself 66 times along 545 base pairs. It also showed variations from one individual to another. Most significantly, these variations were observed in humans as well. This made it ideal to be developed into an investigative probe to identify individuals. Once this repeating fragment was isolated, it was coated with a radioactive substance called P32. When it was exposed to other DNA samples, the radioactive GATA segments attached themselves to their complementary partners. If there were any variations between different samples, they would show up on an X-ray film as different lengths of radioactive bands. The probe was called BKM after the banded crate. And in 1988, it made India only the third country in the world to have its very own DNA fingerprinting probe. It was first admitted in the Madras High Court to solve a paternity dispute, but it wasn't immediately accepted. It would take another paternity case in Tellicherry, Kerala, for the first DNA fingerprinting evidence to be upheld in an Indian court. Since then, DNA fingerprinting has come a long way. This is CDFD, the Center for DNA Fingerprinting and Diagnostics. It's where the country's most difficult cases are cracked, all using the detective properties of DNA. It's used to identify suspects in criminal cases. It resolves paternity disputes. It can identify disaster victims whose bodies are beyond recognition. It has even found great use in preventing wildlife crime and food adulteration. It's also used to diagnose prenatal genetic disorders. And where detectives once took 2 months to conduct DNA fingerprinting, today it can be achieved in a matter of days.
DNA technology has reached a level of maturity where everywhere in the world it is referred to as the gold standard of forensic medicine. We reach levels of probability of a match between an individual and a sample that are far more than the number of individuals on the planet, by more than a factor of 1,000,000.
So how do DNA detectives investigate their cases? They need neither criminal records nor eyewitness reports. All they need is a sample to extract DNA from. This can be extracted from just about any biological source. It can even be obtained if the individual is no longer present to provide it. Blood, saliva, semen, skin, hair, teeth, and bones are all powerful sources of DNA, and even small amounts can reveal a lot. But each sample needs to be handled differently in order to extract DNA. They are put through different rigorous procedures to extract the truth-telling molecule.
The way DNA is isolated either from blood or from this is you break open the cells so that the nucleus comes out, and by using different chemical reagents, and then you isolate the DNA. The decalcified bone powder is now mixed with forensic buffer and phenol chloroform for isolating the DNA. After mixing these components, then they are centrifuged to separate both the aqueous and organic phases. After centrifugation, we get two phases. The aqueous phase contains the DNA, and the organic phase contains all the cellular components.
Once the DNA is separated from other cellular debris, it can be studied more closely. In the lab, simple tandem repeats, or STRs, are obtained by adding restriction enzymes to the DNA sample. Just like a scissor, these restriction enzymes cut the strand into STR sequences. These STRs are then amplified or increased in number by millions in a procedure called polymerase chain reaction or PCR.
A real breakthrough in forensic science came with the discovery of polymerase chain reaction. You know, the biggest problem of forensic science was to get enough DNA from remains and intact DNA. Those deficiencies were overcome by the technique of polymerase chain reaction, for which Nobel Prize was awarded.
Once PCR amplifies the amount of DNA, it's time to study its STR fragments more closely. After all, they are the real DNA fingerprints of the individual. This is done through a process called electrophoresis. First, the sample is dyed into a distinctive color. Then it is added to a gel called agarose. When electric current is passed through this gel, the DNA begins to split into fragments. Depending on molecular weight, these negatively charged fragments move towards the positive poles. Now the plate is put under ultraviolet light, where the dyed fragments of separated DNA will show up as bands. This sheet of DNA bands is what makes us distinctive, even in a crowd of millions. All that needs to be done now is compare the number and frequency of bands against other samples in order to determine a connection. In 1992, this technique was used to solve the sensational assassination of Rajiv Gandhi by linking blood sample DNA found on site with that of the suspects Thanu and Sivarasan. More recently, in 2012, a decades-long legal dispute was scientifically resolved when DNA fingerprinting proved paternity between politician N.D. Tiwari and son Rohit Shekhar. Today, DNA fingerprinting technology is at its most advanced. However, there are still challenges, especially when the source of DNA is either too little or too damaged. Just such a challenge was presented to CDFD by the Gujarat High Court. In the aftermath of the 2002 riots, mass graves of unidentified bodies had been discovered. Samples of the skeletal remains were sent to Hyderabad for DNA analysis and positive identification.
That was important for two reasons. One, it allowed the investigating authorities to establish that this was indeed a mass grave related to the 2002 riots. The second, it was important from a technical point of view because it challenged us in terms of how we could apply DNA technology to decomposed and degraded remains.
Then, in 2010, CDFD faced its biggest challenge yet. A devastating air crash along a steep slope of Mangalore's airport had claimed over 150 victims. Many bodies were beyond recognition, making positive identification extremely difficult. Once again, DNA stepped in as detective. Currently, CDFD's research teams are working on developing technologies where DNA is either in short supply or badly degraded.
To get a successful profile from those degraded samples is a challenging exercise. So now we are looking at different technologies like single nucleotide polymorphisms to help in human identification.
Single nucleotide polymorphisms, or SNPs, are the most common genetic variations found between two individuals. Here, there may be a difference of just one nucleotide in a specific stretch of DNA. This single nucleotide variation can help link an individual to a sample, even if the available DNA is very less in amount. But DNA technologies don't just solve headline-making cases. They can also bring comfort to families grappling with crippling diseases. For many families afflicted by genetic disorders like physical and mental developmental issues, heart disease or cancers, etc., DNA fingerprinting can help cope with the situation and plan for the future.
All these genetic disorders can occur throughout, right from birth of the child to an adult, and whenever they have a problem, either they consult us directly or they are referred to us by a physician. So then we examine them and we decide what test needs to be done, and then these genetic tests help us to identify the reason.
We can counsel the family, and if they wish to have one more child, obviously the parents are worried, will that child also have this error? Will that child also have this disease? How will we be able to cope with it? So before a child is born, when the mother is pregnant, we are able to do certain tests to ensure or to check whether the unborn fetus, the unborn child, has that error or not. And if that error is there, then the parents can be counseled, would they like to have the child or undergo an abortion. So a therapeutic abortion as it is.
A single molecule of DNA, a myriad of complex cases. As a life-giving molecule for all things in nature, its purpose is much bigger than just human dilemmas. And so scientists at CDFD turn to DNA to safeguard one of India's signature products, basmati rice. This fragrant, flavoursome and elegant grain is a celebrity in the rice kingdom. Only grown in this part of the world and highly sought after by many, basmati exports are often victim of a terrible crime, adulteration. Luckily, DNA detectives are there on the scene.
We identified some of the DNA markers which were specific to basmati rice. Using these specific markers, we can actually detect non-basmati rice in the basmati rice samples. So adulteration level detection can be as precise as 1 to 3% level. When we presented this protocol, and also we published these protocols, and we also have a patent on this protocol.
Most recently, DNA fingerprinting has moved on from protecting flora to protecting fauna. Conservation groups around the world and in India are increasingly turning to DNA to fight crimes like poaching and illegal trafficking of endangered species. The wonder molecule of DNA is not just an accurate detective, but also a versatile one.
Now the question is, what is going to be the future technology? Now latest development which is taking place from genomics to phenomics. You have a DNA scene of crime. Can you sequence the DNA and make the picture of the criminal who has committed the crime? So you don't have to go anywhere. The DNA can tell you the whole story.
Nearly 30 years after first being used in forensics, DNA technology in India has come a long way. It has cast its lens beyond sensational court cases to encompass a higher goal.
20 years ago, when the technology was first introduced, there would be questions in court discussing, particularly from the defense lawyers, discussing, is this technique valid enough? Is it accurate enough? Those questions no longer apply. Future of the technology, I would say, is really in expanding its use within India. With a very conservative estimate, we are doing less than 1% of cases that needs to be done, right? And I think our goal will really be to expand.
With these challenges to meet, DNA detectives forge ahead with renewed vigor. In the coming years, they hope to tackle a far wider variety of complex mysteries. Thanks to the double helix, many more case files will then be conclusively closed. If you'd like to share your feedback on today's program, please send your suggestions and comments to Vigyan Prasar, C24 Qutub Institutional Area, New Delhi 110016, or you can mail us at info@vigyanprasar.gov.in.